Hello students, my name is Alok Semwal and in today's lecture I am going to discuss about analytical method validation as per ICH guidelines. In analytical chemistry, qualification of equipment is a prerequisite for validation of the process in which the equipment is being used, and calibration of various measuring devices is an important part of qualification. Various measuring devices include various types of glassware and instruments used in analytical chemistry. Calibration of these devices is very important as we are dependent on these instruments for data collection. If the glassware and instruments used in any type of data collection process are not calibrated, then the data obtained will be under a big question mark. So before understanding the definition of calibration as per ICH, we will understand for what ICH stands for. ICH, which is the International Council for Harmonization of Technical Requirements of Pharmaceuticals for Human Use, is unique in bringing together the regulatory authorities and pharmaceutical industry to discuss scientific and technical aspects of pharmaceuticals and develop ICH guidelines. Since its inception in 1990, ICH has gradually evolved to respond to increasingly global developments in the pharmaceutical sector, and these ICH guidelines are applied by a growing number of regulatory authorities. ICH's mission is to achieve greater harmonization worldwide to ensure that safe, effective, and high-quality medicines are developed, registered, and maintained in the most resource-efficient manner whilst meeting high standards. So according to ICH, calibration is the demonstration that a particular instrument or device produces results within specified limits by comparison with those produced by a reference or traceable standard over an appropriate range of measurements. Following definition defines that according to ICH, calibration is the demonstration that if a particular instrument or device produces results within the specified limits or not. Also, the data obtained is verified by comparison with the data from reference standard within an appropriate range of measurements. A standard is considered as a repeatable, agreed and documented way of doing something. Standards generally contains technical specifications or other precise criteria designed to be used consistently as a rule, guideline, or definition. Standards results from the collective work by experts in a field. For example, in pharmaceutical sciences, IP, which is Indian Pharmacopoeia, BP, British Pharmacopoeia, and USP, United States Pharmacopoeia, are used as reference standards. Hierarchy of standards. There is a three-level hierarchy of physical measurement standards which always remain of prime importance. These are primary standards, secondary standards, and working standards. As the name suggests, primary standards are made to the highest metrological quality, and that is why they are a definitive definition of the unit of measurement. Next one, secondary standards are calibrated by the help of reference to a primary standard. The third one, working standards, are used for the calibration purpose. Generally, calibration of commercial and industrial equipments were done by using working standard limits. Validation as per ICH. Since the generation of ICH, Q2 was the major guideline for the validation of analytical methods. The content of this guideline was previously included in two separate guidelines coded Q2A and Q2B, which were unified to the Q2R1 guideline in November 2005. ICH Q2R1. Prime objective of the ICH Q2R1 guideline is validation of analytical procedures. Objective of validation is to demonstrate and check that if the analytical procedure is suitable for the intended purpose or not. In the analytical method validation, tabular summation of various characteristics is done for the identification, control of errors, and various assay procedures are also performed. Now a question may arise in everyone's mind that why analytical method validation is important. So analytical method validation is very important for ensuring quality control. It verifies and confirms the system suitability. System suitability is the verification of any instrument for any particular method. Analytical method validation is also important for the registration application process of any finished product or method, and it is also essential for submission of report to compendium. Compendium is a directory or a reference book, for example, Indian Pharmacopoeia. Types of analytical procedures to be validated. There are various analytical procedures which needs to be validated in relation to produce accurate results. These methods include various identification tests, quantitative tests for checking the impurity content, various limit tests for the control of identified impurities, and finally quantitative tests for the active moiety in samples or any drug substance or drug product or other selected component or components in the drug product. Before method validation, there are various considerations which must be followed to define a method development goals and acceptance criteria. These include suitability of instrument. Suitability of the instrument is important for ensuring the status of qualification and calibration of instruments. Next one is suitability of materials. This is required for the status of reference standards, reagents, etc. Next, suitability of analyst is for ensuring the status of training and qualification records of the analyst. And last one, suitability of documentation is also confirmed for the suitability of written analytical procedure and proper approved protocol with pre-established acceptance criteria. All of these criteria enables us to determine whether a method has developed in the way you need it for the control of product quality. Various methods that require validation documentation are chromatographic methods, spectrophotometric methods, capillary electrophoresis methods, particle size analysis methods, dissolution methods, titration methods, and automated analytical methods. Analytical method validation. Validation of an analytical method is the process by which it is established by laboratory studies that the performance characteristics of the method meet the requirements for the intended analytical applications. So analytical method validation is the process of documenting or proving that the performance characteristics of the method meets the requirements for the intended analytical applications. Typical analytical performance characteristics used in method validation are specificity, which is also known as selectivity, linearity, range, accuracy, precision, detection limit, quantitation limit, robustness, and system suitability testing. First one is specificity. Specificity denotes the extent to which a diagnostic test is specific for a particular condition or sample. By the use of few specific methods, we can estimate the actual value of the analyte in presence of several other components which may be present in any sample. Various types of identification tests, assay, and impurity tests are performed in the following type of determination method. By the use of specificity, it is possible to determine whether any sample contains impurities or not. Next analytical performance characteristic is linearity. The term linearity refers to the property of scaling. Linearity is a type of mathematical relationship that can be graphically represented as a straight line graph. Linearity of an analytical procedure is its ability to obtain test results which are directly proportional to the concentration of analyte in the sample. Linearity should be evaluated by visual inspection of a plot of signals as a function of analyte concentration or content. For example, linear graph between concentration versus absorbance is obtained by ultraviolet spectroscopic method, and the graph can be used for the determination of concentration of unknown sample. For the establishment of linearity, a minimum of 5 concentrations are recommended. Next performance characteristic is range. The range of an analytical method is the interval between the upper and lower limits that have been demonstrated to be determined with accuracy, precision, and linearity. For example, upper and lower concentrations of analyte in the sample. This range will be the concentration range in which the linearity test is done. In the analytical method validation, a specified range is generally derived from linearity studies, and it depends on the intended application of the procedure. Next characteristic is accuracy. As the name suggests, accuracy is the degree of agreement of test results with the true value, or the closeness of the results obtained by the procedure to the true value. Accuracy should be established across the specified range of any intended analytical procedure. For the determination of accuracy, it is recommended to have minimum of 9 determinations over a minimum of 3 concentrations levels fulfilling the specified range limits. Accuracy is mandatory in case of various assay procedures and in the quantitation of impurities. Next one is precision. Precision of a method is the degree of agreement among individual test results when the procedure is applied repeatedly to multiple sampling from a homogeneous lot. Precision denotes reproducibility or repeatability of results. It is usually expressed as the standard deviation or relative standard deviation of any series of measurements. Precision can be performed by assaying sufficient number of aliquots of homogeneous lot, and after that calculating statically valid estimates of standard deviation or relative standard deviation. Next is detection limit. Detection limit of an individual analytical procedure is the lowest amount of analyte in a sample which can be detected but not necessarily quantitated under the stated experimental conditions. So the detection limit is a crucial factor in determining whether a molecule can be identified and quantified within the acceptable limits. Here, quantification is not possible in all the methods using detection limit. Determination of detection limit is based on either visual examination or signal to noise ratio. Next is quantitation limit. Quantitation limit or quantification limit denotes the lowest amount of analyte in a sample that can be reliably measured by a particular measurement procedure by following precision and accuracy parameters. All analytical techniques have their limitations on estimating analyte concentrations, and there are various representations of the quantitation limit like LOD and LOQ. LOD stands for limit of detection, while LOQ stands for limit of quantitation. Limit of detection is defined as the lowest quantity of the analyte substance that produces a signal at least 3 times the average noise level of the detector. Here is the noise level, and this peak A denotes limit of detection. Limit of quantitation is the lowest concentration of the analyte that can not only be detected but can be quantified within defined limits of certainty. Usually, limit of detection and limit of quantitation refer to limits associated with 95% probability of obtaining a correct result. Next parameter is signal to noise ratio. Signal to noise ratio defines the ratio of an desired analytical signal to the mean noise level for a given sample. It can be used to determine the influence of noise level on the relative deviation of a measurement. For example, in this diagram, following peaks are denoting the unnecessary signal, which denotes noise. Next terms are robustness and ruggedness. The terms robustness and ruggedness refers to the ability of an analytical method to remain unaffected by small variations in the method parameters. Ruggedness of an analytical method is the degree of reproducibility of test results obtained by the analysis of the same sample under a variety of conditions, such as different laboratories, different analysts, different instruments, different lots of reagent, different assay temperatures, different days, etc. Robustness denotes the ability to withstand or overcome adverse conditions or rigorous testing. Robustness denotes property of an analytical procedure to remain unaffected till a defined limit because of careful variations in the method parameters, and provides an indication of its reliability during normal uses. The evaluation of robustness should be considered during the development phase and depends on the type of procedure under study. Next characteristic is system suitability testing. System suitability testing is generally used in laboratories to ensure that the complete analytical system, which include instrument, reagents, columns, and analysts, are suitable for the intended application or not. System suitability testing is an integral part of the various analytical procedures. Following table denotes recommended validation characteristic of the various types of tests under analytical method validation. These includes accuracy, precision, specificity, detection limit, quantitation limit, linearity, and range. In the following table, minus symbol signifies that the characteristic is not normally evaluated, while plus denotes that the characteristic is normally evaluated. 1 is used in cases where reproducibility has been performed, but intermediate precision is not needed. Number 2 denotes lack of specificity of one analytical procedure, which could be compensated by other supporting analytical procedure. Number 3 may be needed in some cases. Following are the references of this presentation. Thank you.